Hope all of you are doing great out there. I'm Ernest, and I'd like to welcome each and every one of us to my channel of Ernest Sciences and Mathematics TV channel. In my discussion today, I'm going to look at coordination by one of the, of course, important ligands which we had looked at in our previous videos. And this is going to be assisting us try to pinpoint specifically what type of amino acids is going to be bonding with a specific metal ions. And this is going to be, of course, dependent on what we refer to as selectivity. Now, in a way, we are going to be giving a general example of this specific kind of a concept in the tail end of this particular discussion in terms of perhaps an illustrative example. Now, from our last discussion, we have decided that basically the metal ions are going to be combining with what we refer to as, of course, complexing agents. And this we said, these specific complexing agents are referred to as ligands. Now, we had introduced the concept of a Lewis acid and a Lewis base in these specific two sets of compounds, or ions and, of course, compounds. And this gave us a way in which we can give a general equation for the combination of this specific acid and, of course, a Lewis base to form this coordinate covalent, of course, product which we can give in terms of or this is specific representation which has been given in terms of that specific coordinate covalent bond given as such. And this gave us a general way in which we can consider the combination of this, of course, metal ion and a ligand to form a complex compounds. Now, in a sense, I said that we're going to have metal ions combining with a range of course, these ligands to form a complex which can be given in terms of this specific representation. And this is going to be assisting us in representing various other aspects of this specific combination of complexes, which are going to be particularly biological ligands, which are going to be joining with the metals in order to form these complexes. Now, we shall have specific names which we are going to be assigning to this specific range of ligands. And in our last discussion, I basically mentioned a few of these ligands which are going to be, of course, important biological ligands, whereby we give an example of these specific ligands as proteins. So I've mentioned proteins as the important biological ligands, and peptides, which we said perhaps particularly a combination of two amino acids and more, which can be given in this specific case. And we said that among those important biological ligands is going to be macrocyclic. So this was macrocyclic ligands, which we gave in terms of crown ethers and, of course, porphyrins. And lastly, we had talked about the concept of this specific third case of nucleobases, which I give an example of this specific illustration using the figures for the ribose sugar, for the dioxy and the ribose sugar, and the difference between the two. Now, in this specific case, these ligands are going to be coordinating with these metal complexes depending on a specific preference, and therefore this is going to be requiring us to try and, of course, characterize the various kinds of ligands which we're going to come across. Now, in this specific discussion, we're going to look particularly as one of the classes of this important, of course, biological ligands which are going to be involved in coordination, and these are going to be proteins. Now, in a sense, I've given a list of some of the amino acids which we're going to come across, and some of the metal ions which are going to be preferred in terms of forming these specific complexes. And I'm going to use this one along our discussion in trying to, of course, elaborate on this specific case. Now, if I said generally that we can, of course, divide this kind of course, ligands, proteins in terms of three divisions. And these are going to constitute a primary, secondary, and, of course, a tertiary type of proteins. Now, we shall elaborate on this specific classification depending on their basic structures given as such, which is basically going to be assisting us. So this is going to be a classification of proteins. So this is going to be generally assisting us in trying to, of course, identify which kind of ligands basically this specific type of proteins are going to be coding with in order to form these coordination complexes. And I said we shall have this specific example of primary proteins. And this, in terms of their structure, basically are going to be amino acid sequences which are going to be formed. And of course, these are going to be consisting of a linear sequence of amino acids, if we can just define it in a summary form. So this is going to be linear sequence of amino acids. So I shall be using this specific, of course, notation for amino acids because of our space, but in case I use this specific notation, then therefore it's going to be referring to amino acids. So basically the first classification of primary proteins is going to be consisting of a linear sequence of amino acids, and we can just give a general example of this specific formation, which is going to be consisting of this amine group joined to the rest of this specific form, and this is going to be consisting of this other center that is going to be given in terms of this, of course, a section for the C terminal given as such, and therefore this is going to be forming, of course, such a kind of a linear sequence of amino acids which can be given in terms of primary proteins. Now we can as well illustrate this one using this specific example, whereby we're going to have these ones represented as that, whereby we shall have this one, of course, given as a sequence of amino acids, which you can give in this specific format, perhaps for just illustration purposes. So I'm just going to have this one given as such, whereby these specific dots are going to be representing the amino acid sequence, and therefore we can just give this specific definition of structures which are going to be primary type of amino acids that consist of amino acid sequence, which can be given in this specific case given as such. Now, so the second part of classification of this specific proteins is going to be secondary, of course, proteins. And in this particular case, we're going to look at what we refer to as the alpha sheets or the alpha helices, and of course the beta sheets, which we have come across, especially in our illustration on the concept of DNA and RNA. So in this case, we shall have this one as a classification number 2, which is a secondary proteins. And in terms of a shape, I've said that this can be classified in terms of this specific interaction between peptide chains, and this can be given in terms of a beta sheet or alpha helices, which I can just give in terms of this specific illustration. So I'm just going to use these color codes to differentiate between the two, and we're going to have this one perhaps given as such. So this type of proteins which are going to be classified as secondary proteins are going to be local foldings, which are going to be forming shapes like the two which I've given on the board. And this is going to be forming a set of repetitive patterns which we can give in terms of this specific alpha helices. So we talked about the concept of a helix, which is going to be formed by these DNA strands. And of course, we said about the concept of this specific beta sheets, which are going to be formed as well for these two specific types of secondary classification of proteins. Now, this is going to be, as well as I said, the interaction between these peptide chains which I've given. And we had mentioned earlier in some of our videos on the concept of various elements which are going to be playing a fundamental role in biological systems, especially of course, element of hydrogen. And I've mentioned that one of the functions of a hydrogen, apart from it being structural, giving a structural form to the this specific ligand of water, it's as well going to be providing what we refer to as hydrogen bonds in this particular structure for the nucleic acids. And this is going to be one of the, of course, applications which we had mentioned. And in a sense, we're going to see that this specific kind of peptide chains are going to be held in shape by this form of a hydrogen bond, as we had mentioned from our previous videos. And this is going to be forming a second set of what we refer to as a classification, of course, of proteins. So we shall look at the third one, which is going to also assist us try to differentiate between different types of proteins for the three categories, which is going to be, of course, referred to as a tertiary. So this is going to be a tertiary proteins given as such. So the difference between primary, secondary, and tertiary, so for the first one, we said that this is going to be a linear sequence for the first one, which is a primary. The second one is going to be formed by, of course, an interaction between peptide chains, which is going to be given in terms of alpha, of course, helices and beta sheets which I've given on the board. And these are going to be held together for the secondary, of course, type held together by the hydrogen bonds as I've mentioned. Now, for the tertiary set of these specific bonds, we're going to see that these are going to be foldings or overall, of course, protein foldings or arrangement of these specific units. And of course, these are going to be determining the various shapes which we're going to come across. So perhaps we can give this one in terms of an illustrative example as well, so that we can differentiate between the two. So in case we have these specific protein sets which are going to be formed, so they're going to be forming different folds which we're going to come across, which can be given perhaps in various forms given as such. And these are going to be held together by using specific, of course, a set of bonds given as such. So in this particular set of proteins, we're going to have some coordination centers which are going to be providing for the particular donors, which we said are going to be giving us coordination centers for metals. And this is going to be assisting us in trying to form, of course, the complexes from these three specific categories of proteins. Now, for the first one, I've given an example of this specific set of linear, of course, set of these specific proteins. And in our last discussion, we had decided that we can give an example of course, a protein which is going to be coordinating with various sets of metal centers, like, of course, the crown ethers and, of course, the ones which I had given for Pharynx. And this is going to be a complex compounds which are going to be having such a kind of centers which we have mentioned. Now, in a sense, if we look at an example like a cysteine, which I've mentioned in some of our videos. So this is going to be an amino acid that is going to consist of this specific set of course, atoms and molecules given as such. So we shall have this one given in terms of NH2 given as such, and we shall have. So for this kind of a molecule, which is an amino acid, basically it's going to be a forming bond using what we call the disulfide bonds, and this is going to be a sulfur-sulfur bond that is going to be achieved from the thiolate, of course, the thiolate sections which we can give in terms of this specific S negative given as such, and that is going to be forming what we refer to as the kind of bonding for such a kind of course, a molecule. So this is going to be a thiolate center that can be given in terms of this particular representation, whereby we say that in this specific case, then therefore we're going to have a similar section of this particular bond being given on this other side, which is going to be, of course, forming another coordination center for this, and the two are going to be forming, of course, what we refer to as a peptide being given as such. So we can consider the case of different types of compounds which are going to be coordinating and forming a range of products. And as we have said, this specific coordination is going to be, of course, characterized by a specific kind of what we refer to as strength. And this case is going to result in the selectivity of specific metal ions which are going to be bonding with certain types of ligands. So in this case, I've just given an example of a table on my right side of the board to show what types of proteins are going to be coding or coordinating with various types of metal ions. And these are going to be giving us a different, of course, complexes, as I've said. So this is going to be referred to as affinity. And we're going to come across these specific terms, and we've talked about perhaps the electron affinity and some of the related concepts from our previous discussions. So the affinity of individual amino acids, which we're going to come across, which basically refers to the strength of interaction at a single sites between two species. And this is going to be characterized, characteristic in a way, and therefore it's going to be resulting to a unique sense of what we call selectivity. So this is going to be characteristics. So the affinity, which is the strength to form, of course, or the strength of interaction at a specific site between the two species, perhaps for the amino acids, and of course the metal ions are going to be forming particular bonds which are going to be characteristics. And this is going to be leading to the concept of selectivity, which we said due to the difference in this specific affinity. And therefore we are going to be, of course, using a unique, of course, procedure for us to determine which metal complex is going to be formed. And this is one of the ways in which we can, of course, determine this is by the use of what we refer to as a metal complex formation constant. Metal complex formation constant. So metal complex formation constant is going to assist us in trying to determine which specific amino acids is going to be coding with that other metal ions given as such. So we can give this one in terms of a general equation of N positive plus amino acid, and this is going to result in the formation of a complex of a metal and amino acid given as such. But in between here, we shall have this specific metal complex formation constant given in terms of a Kf. And this Kf is going to be determining which kind of course, metal is going to be bonding with what kind of amino acids. And this is going to entail the concept of selectivity. In this case, we can see that if we have an amino acid of this specific, of course, nature of histidine, and this is perhaps my pronunciation might not come out correctly. So the first one we're going to have is histidine given as such, and this is going to be bonding with the zinc 2 positive, copper 1 and 2, and then iron 2 ions given as such. And the second type of amino acid which we're going to look across is methionine. So methionine is going to be bonding with this specific range of metal ions which are iron 2, iron 3, copper 1 and copper 2 respectively. So we shall have two sets of this specific, of course, amino acids which are going to be glutamic acid and aspartic acid given in this specific case. So this is going to be glutamic acid and aspartic acid given as such, and they are going to be bonding with iron, of course, iron 3, manganese, zinc, copper, and magnesium given as such. So kindly check on this specific, of course, for clarity. I'm also going to check on the same in case I find a different, of course, oxidation state. I'm going to be mentioning that one in our comment section. So this specific glutamic acid and aspartic acid amino acids are going to be, of course, joining with those specific ions and forming specific complexes. So the last one is going to be cysteine, which I've basically given in terms of representation of a structure. And this is going to be coding or joining with the zinc 2 positive, copper 1, iron 2, molybdenum 4 and 6, and then nickel 2 ions given as such. And this is going to be forming a different range of compounds which we're going to come across. And in our second video, we're going to look at a specific example of a carbonic anhydrase with the zinc, which is going to assist us try to show how sometimes these metal complexes can combine with water to form resting states of these specific complexes. And this is going to be forming a relevant transition in some of the reactions which we're going to come across in our various discussions. So join us in our next video as we're going to look at this specific example, and that's going to be forming our last bit of the coordination by proteins of this specific formation of various complexes. So that is it for now, guys. Bye, God bless you. Catch us in our next video.